Where we left off last time was talking about um, the transcription part of it and replication within the cell of DNA and converting it to RNA, right? Now what I wanted to start off with today is a lab technique called PCR, polymerase chain reaction, which uses everything within a test tube to produce or remake the actual DNA replication. Because it's a lab technique and because this is not something a cell does, we actually take advantage of all the different counterparts of it, or the parts of it like the polymerases, the DNTPs, and those primers that we talk about for DNA replication. But instead of having it all done at one time, we use specific temperatures associated with it. For example, to split the DNA into single strands, we use a denaturing temperature of almost boiling temperature, 95 degrees Celsius. Then by dropping that temperature down, it allows for the DNA to anneal back together again. But we don't want the two strands of the single strands to anneal back. We oftentimes just want the actual primers to anneal. And that's what we do at the 40 to 50 degree range. And then we do an extension which allows for our polymerase, which works perfectly at 72 degrees Celsius. And this will actually extend all the actual DNA strands to make the replication happen over and over and over again. Now the reason why we do PCR is we are amplifying DNA. We have a target sequence that we're looking for and we can amplify it over and over and over again. Why would we want to do this in this case? Well, it's not something where you're trying to replicate a whole entire genome at one time. You're trying to actually just replicate a very, very small or specific sequence associated with that DNA. For example, the applications that we look at it, you can imagine you have things like identification, right? Paternity tests. You can do for forensics, right? Looking for people that are specifically there or present within it. You can look for genetic disorders and pathogens, right? Oftentimes it's these things where you have very, very, very small amounts of DNA and you need to amplify a lot of it, okay? Or looking for identification for specific things. You definitely have advantages of using these things. You don't need much sample. It's relatively quick nowadays. You can definitely do it less than an hour. Yeah, but the big negatives of PCR is that you can't distinguish between dead and alive cells. So it just picks up DNA, so it's really difficult to say if something is present or dead or not there. If you were there at one point, then you will have that DNA associated with it. Okay, but it doesn't mean that you're necessarily there at that second. Because you're looking at these dead versus alive, it's really difficult to use this type of application on every single one of these different cases. Okay, and if you want to have something fun too, you can also even do it for genetic testing of your dogs. Right? If you have poi dogs or mutts and you have no idea what they are, you can definitely look at the genetic information, and you can go to this YouTube commercial, which is pretty funny, where you just take a swab, you swab your dog's mouth, and it will tell you exactly what kind of pet makeup that you have of these types of dogs. So it's really interesting to see it. Definitely, it's become mainstream nowadays. You'll see it on TV. Um, all these different types of advertisements nowadays. You can even have your own genome sequence through this different type of programs that they have nowadays. Okay, so for a few bucks, you can do all of your genetic testing that you need. Okay, now we're getting into DNA translation. Okay, so we talked a little bit about polymerase chain reaction, right? That's something you have to understand. It's a lab technique necessary to do so. But you got to look at the different specific RNA translation mechanisms. For example, we need to know tRNA, mRNA, and rRNA, right, along with the genetic code. Okay, and more specifically, we're going to have to understand how to translate this RNA into protein. So we have to understand things like start and stop codons. Okay, and from this, then we're going to start looking at regulation. What an operon is at the RNA level or DNA level, and try to look at what the actual ways of producing or expressing these genes to make functional products. Okay, and one more thing, we have this amino acyl tRNA synthetase. This is just a specialized enzyme that connects the amino acid specifically to that tRNA. Okay, genetic code. Genetic code, we talk about this as having abbreviations of 3 letters, right, to make a codon. And each codon represents an amino acid, right? So we have 20 amino acids, and each one of these codons will represent a specific amino acid. And we do have some redundancy. For example, we'll be talking about termination codons like UAA, UAG, and UGA don't encode for any type of amino acid. okay, they encode for stop codons, which means that these are termination sequences associated with it. There is no such thing as a stop amino acid. It's just something where you have an empty tRNA that gets placed there and it stops the translational process. Okay? For start, you have a specific amino acid called MET or methionine. For this cases, you have a specialized one called AUG, and oftentimes this is the only start codon that oftentimes you'll be familiar with. And this is the initial starting amino acid. For example, every single gene that gets translated that has an AUG, right, is always going to start with the MET amino acid. So here is your genetic chart, right? So you don't have to memorize this if on an exam and it asks you to translate something, right? You'll have something similar that you can translate it with, but you should be very familiar with looking at how to find what each specific codon represents which amino acid. Okay. Here's your start codon, here are your stop codons, right? But if you look at, for example, proline, right, you can see it has multiple different types of codons that is going to represent that proline, right, like arginine, but histidine only has these two specific ones. So make sure that you look at the three-letter combinations to represent with its corresponding amino acid. Okay, for example, right, you can take a DNA strand, right, that I give you here as an actual trial, right, ATG, GGA, AAT, GCG, GAC, CAT, and then you can actually do replication, right? You can tell me what the actual replication of that strand is going to be. So you better make sure it goes in the DNA fashion. Yeah. From this, you can even tell me what it's being transcribed, right? So you can be able to convert this into RNA. And with that RNA, you can do an RNA translation from it, right? So you can tell me what the actual sequence of the amino acids is going to be to produce something, some type of peptide. So make sure you're able to do this. Again, these are great exam questions to be able to give you. So you can do this kind of switching between DNA all the way down to the amino acids. Okay, so how does this all happen? Right, so we got to talk about a little about RNA translation. So you have tRNA, right? The one component of doing translation, and this is the transfer RNA, right, which connects the amino acid directly to that ribosome and the mRNA. Okay, each amino acid has to be joined to that transfer RNA by a specific enzyme, and that specific enzyme is called an amino acyl tRNA synthetase. Okay, you have 20 different synthetases which match with 20 different amino acids, which means you have a very specific interaction between the enzyme and that amino acid. Okay, so it's not being able to do all of them. You have one enzyme to do one connection of that specific amino acid. And once it does that, it's going to make that activated amino acid. So in that case, you're going to have an activated tRNA with that amino acid adjacent to it or stuck to it, and it's going to bring it to that translational site. Okay, so here you go with the beginning part of it, where you have an mRNA strand, right? And here's your ribosomal unit. Okay, it comes together at the AUG site or the start site. For ease purposes, let's just say that with the start site, the ribosomal subunits come together, and when they do, you're going to get that first initial tRNA, which brings the methionine to that site. So it connects it to that tRNA, and it puts it in right into that first spot. Okay, and initiates the whole entire translational process. As you get that first codon get placed there with the AUG, the next set of 3 is going to give you the next codon, right? The next amino acid. In this case, it's going to be leucine, which matches up perfectly. Okay, get a peptide bond, release the 2 with the leucine and methionine, which then exits the first initial tRNA, right? And so now you're building your peptide off of each one. So as the ribosome goes down and reads every codon after that 3 of AUG, you're going to basically start making a longer peptide, right? So here's your growing peptide. So every time a new one comes in, you can connect it. So as it keeps going, right, and it puts this all together, and you get a very long strand of amino acids. You can see here that oftentimes a question I get is, do you have only one methionine per peptide? Well, no, you can have multiple methionines. Okay, it doesn't really matter. It is an amino acid, but usually you always will begin with a methionine as that start codon. Okay, Now, how does it stop? The way it stops is having it come in contact with a stop codon. That stop codon here is the UGA in this case. Okay, and this is going to stop the translational process. Again, we talked about not having an actual specific amino acid associated with it, but it's going to be an actual codon which doesn't allow for anything to occur, which means it's an empty tRNA that comes to that site, and nothing is going to be able to bind to that growing peptide, and everything is released and falls apart. And once you have that, you have your new protein there, and that gets actually folded up, and then you have your actual protein or enzyme or structural thing that you need for the cell. Okay. As you can see, it doesn't just happen with one strand happening at one time. You can get multiple amounts of mRNA there, right? All being translated, okay, produced by all these ribosomes to make these peptides to make it much more efficient. Okay, so this thing is not just happening at one time. You can have multiple amounts of it happening at every single moment, as long as you have that DNA strand right here that's going to get directly transcribed into each one of these strands. Okay, and as that's going, you're going to have reading of these ribosomes off of it to make those specific peptides. So how does it know when to transcribe these certain genes? Or expression and regulation plays a big role. Okay, and oftentimes with this one, we use the classic lac operon to understand how this thing is happening. So with lac operon, there's things that you have to understand. What is an operon, right? What are the actual units associated with it? And how these things are being expressed or induced or repressed during these actual moments. So an operon consists of three components, and I'll show you a figure in a little bit that shows the three parts. But you have an operator, a promoter, and structural genes. Okay, the two major things that you have to understand is that the operator and promoter are not transcribed. Okay, they're just non-coding pieces of DNA. So this operator is a region of DNA adjacent to the structural genes that controls the transcription, tells it when to turn on or off, or initiates this whole entire process. The promoter begins the whole entire thing, and the structural genes is what you're actually transcribing, right? That's that protein sequence that you're trying to make. Now when we talk about operons, you can have these things as constitutively expressed, which means they're always on, okay? Or you can turn them on through induction, or you can turn them off through repression. And oftentimes these are all undergoing certain types of regulation to make sure that these genes are not always on all the time. Why would you want some genes on all the time? Well, you can imagine these are not going to be expressed at high levels, but definitely at a fixed rate, because maybe these are housekeeping genes, right? These are ones that you want to have on all the time to make sure the cell is working properly. Okay, so here's your cartoon figure of your structure of your operon, right? So here's your DNA that you have here, and what these segments of letters are showing you is the specific areas of what you're looking at. So for example, you have your operator, okay, right adjacent to the structural genes that you're trying to produce. So you're going to make gene Z, gene Y, and gene A, right? And here's your promoter sequence that initiates the whole entire transcription of everything. Yeah. The thing that I'm trying to label here that do not transcribe anything is that these are non-encoding regions, these control regions. They are just information sequences to tell when to turn on or off these structural genes. For example, when you look at the lac operon means that these are genes that are turned on only in the presence of lactose. Okay? So if lactose is present within the actual cell, or is present, these genes are going to be expressed or induced, right? Cells don't like lactose. If there's a choice of a different type of carbon source, it will use it. Okay, so in this case, it always has it off, right? You don't want to have these genes turned on unless there's lactose present or no other carbon source that's present for these guys to use. So in this case, when there isn't any lactose present, you're going to have a repressor protein. And that repressor protein binds to the actual operator sequence, right? And physically blocks the RNA polymerase from doing any type of transcription of genes Z, Y, and A that are necessary to break down lactose. Yeah. So this repressor is always going to be made, and it's always going to be blocking that RNA polymerase. But this is only when there is no lactose. When there is lactose, you want to make sure you have the structural genes necessary to break down lactose So you need to have this induced, right? You need to have it turned on. Lactose, when present, actually causes a conformational change in that repressor. So it changes the structure of it so it no longer can bind to the operator region. And because it can't bind anymore, the RNA polymerase can then go through its actual transcription process and make the specialized enzymes necessary to break down lactose, like these permeases, transacetylases, and beta-galactosidase that are necessary to do this process of breaking down lactose. Okay, and again, the only time you have this present is when there is lactose. Okay, if there's no lactose, these genes will not be made. So a repressible operon is the exact opposite. Okay, it's always going to be on until it's turned off, right? Inducible operon, what we just talked about, it was always off, and then we can turn it on. This is going to be the opposite of it. Okay, for example, when you want something like tryptophan, which is necessary as an amino acid, right? You always want these type of actual synthesis genes to be turned on, so you're always making tryptophan. Okay. So in this case, you have an inactive repressor, right? You don't want it to bind to stop the whole entire process. And so you're going to allow for the RNA polymerase to make all the structural genes necessary to synthesize tryptophan. Okay? But if you have too much tryptophan, it doesn't need this operon to be turned on anymore. You don't need this process to occur, so it can turn it off. Right? How do we turn it off? Well, if there's enough tryptophan, it actually can bind to this repressor, okay, thereby activating it, and then binding to the operator sequence to stop that RNA polymerase from making those structural genes. So in this case, this is a repressible operon, definitely not an inducible one. This works for biochemical pathways too, right? You can imagine with feedback inhibition, or this process of competitive inhibition can stop this whole entire process from happening too. So in this case that we were talking about before, we're talking about gene transcription, right, at the DNA level and making RNA. Here we're talking at the biochemical pathway of looking at how these things can stop enzymes by doing something very, very similar, right? So in this case, we're not talking about something that's inducible or pressible. We're just talking about feedback inhibition, which will stop the whole entire process, right, if you have enough of an end product, okay? And so this will stop oftentimes this whole entire metabolic pathway or biosynthetic pathway to stop it from using the enzymes to keep making it over and over again, or to do this process when it doesn't need to. Now going back to this catabolite repression or using glucose and lactose, it's always really confusing to students to hear why lactose isn't being used. Well, it's really clear when you start doing graphs associated with glucose versus lactose. Okay, so here we had time, right? And this is kind of like your growth curve very basically. And here's your number of cells, right? So over time, when grown only on glucose, the cells love it. They grow really, really fast. Okay, lactose, not so well. It doesn't want to choose, right? You want to choose a milk, yogurt over a big Snickers bar, right? You can imagine having this is not going to be a very, very slow process of boiling it down, but it can definitely break it down. Okay. If you give the cells a choice and you put glucose and lactose in the same media at the exact same time, glucose is only going to be broken down first, okay, and then you get lactose being broken down. It's not going to use it at the exact same time. What they call this is a dioxic growth curve, where you have one first carbon source being broken down or used. Once it's all consumed, it goes back into a lag phase, right? And then goes back into the exponential phase with the next carbon source. In this case, the lactose is used. So when we see this, we can actually see this properties happening when you put both actual carbon sources in. Okay, the way that we see this working is things called the catabolic activator protein, right? The catabolic activator protein, or CAP, is kind of a general protein that helps initiate a lot of these pathways that are not necessary for all the time. Okay, so when there's lactose present and no glucose, this CAP protein will sense the very low levels of glucose, okay, by having increased amounts of cyclic AMP, and it's going to bind to it and activate it. And the reason for this process is that it helps recruit the RNA polymerase to that site of the promoter to allow for transcription to occur. Okay, So it's one extra step that we're learning within this lac operon. But this is only in the case when you have no glucose. Okay, if there is glucose and lactose, you get something completely different, right? Remember we said glucose, when it's present, it's going to use it, right? In this case, if it's using it, you're not going to have very high levels of cyclic AMP. Because you don't have very high levels of cyclic AMP, this cap protein stays inactive. And if it stays inactive, you can't get recruitment of the RNA polymerase, and you don't get any actual transcription, even though you have lactose present, which is inactivating that repressor, because the cell wants to use glucose first. It's a much, much better carbon source. Okay. See, the only time you're going to get transcription of these genes is only when lactose is present and there is no glucose. Okay, so ending this actual lecture, here's the last question, right? All of you know that antibiotics will affect specific characteristics that are only found in prokaryotes, like bacteria, not eukaryotes, like us. From last lecture or from this past lecture, what could antibiotics be targeting that are specific to prokaryotes, and how does it work? So if you have an answer, let me know, email me, or ask me any questions, and I'll try to help you out as much as possible.